Hello students, I am Parna and welcome to Parna's Bioclasses. So, we are studying Unit 3, Cell Structure and Function. Today is lecture 2 of the series and today's topic is about prokaryotic cells. I have tried to cover mostly everything that you need to know about prokaryotic cells, and if you listen to the video lecture with concentration, I'm pretty sure by the end of the lecture you will be confident. So sit back, take out your notebooks, take down some notes and listen to the lecture. Do like, share and subscribe my channel and definitely comment. That matters a lot. So without wasting any further time, let's start. In this lesson, we will learn about prokaryotic cells and prokaryotic cell organization. Prokaryotic cells are morphologically much simpler compared to eukaryotic cells, but they have their own characteristics. The organelles in prokaryotic cells differ from that of the eukaryotic cells. Example of prokaryotic cells includes bacteria, blue-green algae, which are also known as cyanobacteria, mycoplasmas. They lack cell wall. Mycoplasmas are bacteria which lack cell wall and which are the smallest living cell available. And PPLOs. PPLOs are pneumonia like organisms. Earlier, PPLOs were named as PPLO, but nowadays mycoplasmas are the term used for PPLOs. Prokaryotic cells are found in four major shapes. Bacillus, which is rod-shaped. Example of a bacteria which has this shape is Bacillus subtilis. Next shape is coccus, that is spherical. Example of a prokaryote having this shape is Staphylococcus aureus. Vibrio is another shape of prokaryotes which is comma-shaped. Example of a bacteria having this shape is Vibrio cholerae, which causes cholera. And another shape which is available is Spirillum, which is spiral shaped bacteria. Example is Helicobacter pyroli. So we learned that prokaryotic cells are morphologically simpler. They are found in four major shapes, and we came to know about few prokaryotes which are available widespread. Here we have a schematic diagram of the prokaryotic cell and the various structures that comprises the prokaryotic cell. We will start with the outermost covering of the protoplasm known as cell envelope. It is a three-layered structure that comprises of the outermost glycocalyx, then cell wall and the innermost cell membrane. The cell envelope forms the outermost covering of the protoplasm. Then we have the cytoplasm, which is an aqueous gel-like compartment guarded by the cell membrane. It hosts structures like ribosome, chromatophores, mesosomes, and inclusion bodies. Then we come to the nucleoid, which is an irregular shaped region containing the prokaryotic genetic material. Apart from the nucleoid, we have plasmid, which is extra chromosomal segment of DNA. Then we have flagella, Fimbriae and Pili. These are external structures protruding out of the cell. They have their individual specific functions. Now we'll take a look at each of these prokaryotic structure in details. We will now know about cell envelope. It is the outermost covering of the protoplasm of the prokaryotic cell. It is a three-layered structure. The three-layer consists of the outermost Glycocalyx, the layer which is represented with brown color in the picture. Then we have the cell wall, the yellow layer, and the innermost cell membrane, which is highlighted by the color green. These three layers together form the cell envelope. Now we will know about Glycocalyx. Glycocalyx is a structure which is external to cell wall. So it is the outermost layer of the cell envelope. It has two parts, inner S layer and outer mucilage layer. Glycocalyx is composed of many substances, primarily polysaccharides. Glycocalyx gives sticky nature to the cell. Glycocalyx can be of two forms. If it is in the form of loose sheath, very unorganized and can be removed easily, then it is called as slime layer. And in case it is thick, tough, well organized, which cannot be easily washed off, then it is called as capsule. There are many functions of Glycocalyx. First one is adherence to surfaces. As we already saw that it contains mucilage layer, slime layer composed of polysaccharides, it gives a sticky nature to the prokaryotic cell and thus helps it to adhere to different surfaces. It gives protection to the cell against phagocytosis. Phagocytosis is a process by which other cell tries to engulf this cell. So the cell envelope, particularly the Glycocalyx, gives the cell protection against phagocytosis. It protects the cell from desiccation, that is, drying up. It also gives virulence capacity to the prokaryotic cell as it protects the cell from the host immune system. Now we will discuss about the cell wall. Cell wall lies outside the cell membrane or plasma membrane. So, as part of the cell envelope, cell wall lies interior to Glycocalyx and lies outside to cell membrane. It is a rigid, solid covering. In terms of its function, It provides shape to the prokaryotic cell, that is, it determines the shape. It also gives structural support to the cell by preventing it from osmotic lysis. Osmotic lysis takes place when the concentration of solute is different in both sides of the cell envelope. So, if the concentration is less outside, which is a hypotonic solution, or more, which is a hypertonic solution, there is a chance of osmotic lysis of the cell. Cell wall prevents osmotic lysis. Most bacteria can be divided into two groups based on their response to gram stain. The gram staining procedure was developed by the scientist Hans Christian Gram. I would make a separate video lecture on gram staining in elaborate way. So the two categories of bacteria based on their response to gram staining are gram positive and gram negative. Now, this difference of behavior towards gram staining is due to their difference in cell wall composition. Cell wall is made up of peptidoglycan, also known as murine. Peptidoglycans are composed of long glycan chains which are cross-linked by peptide chains. Hence the name peptidoglycan. So this was all about cell wall. Now we will discuss about cell membrane. Cell membrane is the innermost layer of the cell envelope. It is selectively permeable. So what does the term selectively permeable means? That means it is very selective in choosing that which solutes or ions or molecules it will allow to pass across it. It will not allow everything to pass. It will choose what to pass and what not to pass. It is the covering of the cytoplasm. It is made up of phospholipid bilayer. Phospholipids are amphipathic in nature. Amphi means both. So, phospholipid molecules have a polar head group which is hydrophilic and a non-polar tail which is hydrophobic. When these kind of molecules come together, they form phospholipid bilayer which will be arranged in this fashion. It will arrange in such a way that the hydrophilic heads will be outside facing the aqueous part of the environment and the hydrophobic surfaces will be buried inside the membrane. Now, it is not only the phospholipids that are present in the cell membrane, there are also proteins floating in the membrane. These proteins can be of three types. They can be extrinsic, that is, they may lie on the surface of the phospholipid bilayer, or they might be integral, that is, they may be present inside the bilayer, or they may be transmembrane, that is, they span across the phospholipid bilayer. These are the three types of proteins that can be part of the plasma membrane. And this model of plasma membrane was proposed by Singer and Nicholson, and this is known as the fluid mosaic model. Now we will discuss about cytoplasm and the various structures present in the cytoplasm of the prokaryotic cell. Cytoplasm is an aqueous, gel-like compartment. It is guarded by the cell membrane. So the layer of the cell envelope which is in contact with the cytoplasm is the cell membrane. It hosts various structures in the prokaryotic cell like the ribosome, mesosome, inclusion bodies. So now we will discuss about ribosomes. Ribosomes are very small structures present in the cytoplasm. They can be present in two forms. Either they can be fixed, that is attached to the plasma membrane, or they can be free, that is lying in the cytoplasmic matrix. They are membraneless structures. They are composed of ribonucleoproteins. This means ribosomes consist of two things, RNA as well as proteins. Hence the name ribonucleoproteins. Prokaryotic ribosomes are 70S in structure. What does this mean? S stands for Svedberg unit or Svedberg number, which is a measure of the sedimentation coefficient. So, if we try to centrifuge various organelles or structures, sedimentation coefficient or Svedberg unit is the number that determines that which organelle or structure will sediment faster. Heavier the structure, more will be the Svedberg number. So the prokaryotic ribosome has a Svedberg number or Svedberg unit of 70S. It has two parts. The prokaryotic ribosome consists of two subparts. The bigger, larger unit has Svedberg number of 50S and the smaller one is 30S. So prokaryotic ribosome is 70S ribosome which has two subunits 50S and 30S. Please do not get confused. By adding 50 and 30 you will not get 80. 50S and 30S are the sedimentation coefficients of the larger and the smaller subunits, and 70S is the sedimentation coefficient of the entire prokaryotic ribosome. Function-wise, ribosomes are the site for protein synthesis. That is, the process of translation takes place at the ribosome. Translation is a process by which mRNA is used generating protein. Protein is synthesized from mRNA code. This process is called translation. Ribosomes can form polysomes or polyribosomes. And these polysomes or polyribosomes can attach to single mRNA during translation and generate multiple copies of a protein. Now we will discuss about mesosomes. Mesosomes are folded invaginations. They are infoldings of the plasma membrane of bacteria. These mesosomes can be in the form of vesicles, tubules, or lamellae. They help in cell wall formation. As well as other processes like DNA replication, cell division, respiration, secretion. Since mesosomes are infoldings or invaginations, they increase surface area of the membrane. More surface area means more enzymatic content. Hence, more metabolic activities. We will now discuss about inclusion bodies. Inclusion bodies are non-living structures present in the cytoplasm. Inclusion bodies are categorized into three broad categories. Number one is gas vacuoles. These are gas storing vacuoles. They are found in organisms like cyanobacteria, which are also known as blue green algae, then purple photosynthetic bacteria, as well as green photosynthetic bacteria. These gas vacuoles are made up of gas vesicles, and one of their important function is to regulate buoyancy in aquatic photosynthetic bacteria. Regulation of buoyancy means that the photosynthetic bacteria which are present in aquatic environment, they can float and adjust at a depth which has proper light intensity, oxygen and nutrients. So the depth at which it will be floating is regulated by these gas vacuoles. The second type of inclusion bodies are inorganic inclusions. This can be volutin granules, which consists of phosphate. They serve as phosphate reservoir. It can be sulfur granules, which serve as sulfur storage. Also, the third category is food reserve inclusion bodies. So, as the name suggests, these inclusion bodies are storage of food material or organic storage. It can be cyanophycean granules, which are a storage of nitrogen. They store polypeptides. Glycogen granules, which are carbon storage, and PBH granules, that is poly beta hydroxy butyrate granules. So, just to summarize, inclusion bodies are categorized into three major groups. First is gas vacuole, second is inorganic inclusion bodies, and the third is food reserve. In inorganic inclusion bodies, we have the volutin and the sulfur granules, and in the food reserve granules, we have the cyanophycean granules, glycogen granules, and the PBH or poly beta hydroxy butyrate granules. Apart from the structures that we discussed in the prokaryotic cytoplasm, that is ribosomes, mesosomes and inclusion bodies, there is one more structure we should know about, that is chromatophores. Chromatophores are internal membrane system. They are found in purple as well as green photosynthetic bacteria. So these are not present in all prokaryotic cells. They contain photosynthetic pigments like bacterio, chlorophyll, carotenoids, and others. Now we will discuss about nucleoid. Nucleoid is an irregular shaped region. The prokaryotic genetic material, that is the chromosome, is located in the nucleoid as these cells lack well-defined membrane-bound nuclei. The prokaryotic genetic material that is present in the nucleoid is usually single copy, that is a single chromosome, which is double-stranded and circular. This is happening in most of the cases, but there are some exceptions, like in some prokaryotes, they are linear. For example, in Vibrio cholerae. The chromosome that is present in the nucleoid is supercoiled, but they are not associated with histone proteins, which we will see in the eukaryotic cell. The prokaryotic chromosome or genetic material is often referred to as naked. This is because there is no nuclear envelope around it, and also there are no histones attached to it for packaging. The nucleoid is also embedded in the cytoplasm. We already learned about nucleoid. That nucleoid contains the prokaryotic genetic material or chromosome. Apart from the chromosome in nucleoid, there are certain prokaryotic cells which contains extra chromosomal segments of DNA. These are called plasmid. Plasmids are self-replicating, that is, they can replicate on their own. Usually, plasmids are double-stranded, circular, naked DNA, that is not enclosed by any membrane-bound organelle. The replication of plasmid is often referred to as autonomous replication, as the replication of plasmid is independent of the replication of the main nucleoid. Plasmid contain important genes for various factors like fertility factor. Prokaryotic cells containing genes for fertility factor takes part in bacterial mating, that is conjugation. Other important genes that are present in plasmid are the genes for antibiotic resistance. If a cell has plasmid containing antibiotic resistance genes for any particular antibiotic, then that cell will become resistant towards that antibiotic. Also, plasmid contains colicin producing genes. Colicin are proteins that are produced by one prokaryotic cell like bacteria, which can act lethally, which can be lethal towards other bacteria. So all these genes are present in plasmids. Plasmids get associated temporarily with nucleoid forming episomes. Due to the various genes present in plasmids, plasmids are important vectors in genetic engineering studies. We will now learn about prokaryotic flagella. Flagella are locomotor appendages. That is, they are needed for locomotion. Hence, their function is providing motility to the prokaryotic cell. They are thread-like, slender, and rigid. The prokaryotic flagella is made up of single microtubule fiber. When we study about eukaryotic cells, we will see that the case is different in eukaryotic flagella. Flagella consists of three parts: filament, basal body, and hook. The flagellar filament is the longest part of the flagella and it extends from the cell surface to the tip. The basal body is embedded in the cell and the hook is also embedded and it is short and curved. Flagella is made up of the protein flagellin. Flagella consists of a stator and rotor. The stator and rotor together causes rotation type of movement in the flagella. Now we will learn about pili and fimbriae. Pili are elongated, longer, tubular outgrowths. They are made up of the protein pilin. The function of pili is in bacterial mating, that is conjugation. Fimbriae are smaller, bristle-like or hair-like fibers protruding out of the cell. The function of fimbriae is attachment. Fimbriae helps in attachment of the bacteria to solid surfaces as well as host tissues. If you find the video helpful, do like, share and subscribe. It matters a lot.